Thanks for the kind invite, and I want to describe to you today some of our efforts in trying to do cancer genomics to help patients. And the title is Harnessing Genetic Interactions, and I'll explain, to try and advance whole genome, and I emphasize whole genome precision oncology. And these are efforts that are now ongoing in this new cancer data science branch that has just been opened by the NCI. And as you may see, it's the work of two really bright postdocs in my lab, Avinash Das and Ju Sang Lee, with many experimental collaborators. So as you know, the current state in cancer treatment isn't great. Yeah, when I'll say something which is worth listening to.
How can we know?
You know me, about 10% is informative. So, you hear me? Is that okay? As you know, current cancer treatments aren't great and needs to be improved. And one main problem is that they're not effective and selective enough, and the other is that many times we have initial great response, but then regrettably after a year, 2, 3 years, resistance emerges. And I want to describe some efforts that we have been doing trying to combat both these challenges. Now, as you know, there is a lot of hype in recent years about precision oncology, and for those of you who don't know, the basic idea is to sequence about 300, 400 cancer driver genes. Yes, the main tumor suppressors and oncogenes that are known to be involved in cancer and are highly mutated, and to try and find actionable mutations. What are actionable mutations? These are mutations for which we happen to have drugs. Some have been developed specifically, some have been repurposed to treat that. And you probably heard like companies like Foundation Medicine or Tempus, who offer valuable services to do that. But the main problem is that the coverage, the number of patients we can help via these methods isn't very high. Estimates vary, like between 3% to 20% or something like that. And the majority of the patients have many alterations that are occurring not on these cancer driver genes, okay? Via copy number, transcriptional, other alterations. And the question is, can we do something about it? And furthermore, we know from basic science that what happens in cells is very much dependent on the wide context of all the genes in the cells, and not just on these cancer driver genes. So in a way, the current approaches that we offer is like looking for a coin under the lamp. Okay, and the question is, can we look? Can we develop a precision-based oncology treatment that looks on the whole genome of a biopsy from a patient's tumor and tries to do something about it? And I want to show you our first attempts in developing such a method, which, by the way, is now starting to go into various clinical trials in the NCI. And of course, the road from clinical trials to actually help patients is a few years, a lot of money, and remains to be seen. And our approach builds on two kinds of genetic interactions, which I'll describe. One of them that perhaps many of you have heard of, synthetic lethal interactions. The other one is more remote, called synthetic rescue interactions. And I'll explain what we do. So the first part that I'll talk about synthetic lethal interactions, was just actually published like a month ago or something like that. So it's a paper that's available if anyone is interested. And of course, I'll be very happy to talk with anyone who is interested and explain more and so on. And it's based on the idea of synthetic lethality. So two genes, A and B. By the way, stop me anytime, okay? Anything. Genes A and B are synthetic lethal if the knockdown, the inactivation of each of the genes alone doesn't do anything to the cells. But the knockdown of both genes together kills the cells. okay? Now, in 1997 or 8, appeared a paper in Science that had 4 co-authors, 3 of them Nobel laureates, 1 of them just a National Academy member, okay? And they said that if we will find the synthetic lethal interaction pairs in cancer, that would be a huge stride in our ability to treat cancer. Now, I'm asking the Nobel audience, why, if we would just find the damn synthetic lethal interactions in cancer, we would be able, that would be revolutionary in our ability to treat cancer. Come on. Suppose you know someone, yes, sorry. And fantastic, and fantastic. Okay, suppose I know, just say 1 pair, A and B, and I'm taking a tumor that is synthetic lethal. I'm taking a biopsy from a tumor's patient, and I see the gene A is lost, and I happen to have a drug that targets gene B. So bam, at least theoretically, conceptually, I have an amazing opportunity to treat and kill only the cancer cells selectively, right? Because I give the drug, only the cancer cells have lost gene A, I give the drug target gene B, the rest of the cells are theoretically, conceptually happy, and I kill the cancer cells, right? So 20 years have passed, and we haven't yet discovered the synthetic lethal pairs in cancer in human cells. Why? Why are we so lagging behind? The context may affect, you're right, deeper context may affect, but that's minor still. Why didn't we identify the pairs? Excuse me? The experimentalists are lazy? That's your conclusion? What the hell? You don't need to sequence. Just do the damn double knockouts in all types of cancer cells and identify the synthetic lethal pairs. What's going on? The combinatorial problem. Who said that? Yes. 500 million potential pairs. And the experimentalists have done a fraction until today. Okay? Now I'm going to tell you now something shocking. And you won't believe me. We have come with a computational method, which is pretty, in principle, simple, and identifies all the synthetic lethal pairs in cancer directly from the patient's tumors. So it's not even, we bypass the problem, I don't know if you may be aware, but what you find in vitro so many times, even in vivo, is not relevant to human tumors. But we identify them directly. Now, are you listening? I'll explain to you the principle. So simple, so shamefully simple. The principle is the following. Suppose you were talking about public data sets and their value. Tremendous value. So the TCGA, as you may know, is a huge repository, 10,000 patients' tumors. You have all the king's horses and all the king's men, right? The sequencing, the RNA sequencing, some proteomics, copy number alterations, everything. So you have a matrix of 20,000 genes, 10,000 tumors, and by looking on the expression and copy number of each gene, you can estimate the likelihood that each gene is lost in any given tumor. Okay? You with me? Suppose you mine this large matrix, and you identify genes A and B. You do it for every pair, A and B. You identify genes A and B, which are sometimes lost. Okay? They have a frequency to be lost in all tumors, gene A, a frequency lost to be that it's lost in all tumors gene B can easily compute by the gene expression, copy number, mutations, whatever. But you almost never find them lost together again, together in the tumors. Yes, they are lost individually, here and there. But you never, almost never, find them lost together in the tumors. What does that tell you? Come on. What does that tell you? You correct for multiple hypotheses over 500 million pairs carefully, and you still get stratospheric p-values that this is inconceivable for thousands of interactions. What does this tell you? Thousands of pairs. But they are most likely, this is strong evidence that they are synthetic lethal. Why? Why?
Because if one exists, then the other doesn't.
Because if they are lost together, the cells die and you don't see them in your samples. Now, furthermore, you carefully compute the survival effects of the inactivation of each gene individually, and whenever they are lost together, the survival effects when they are lost together. And you find, yes, please.
What's the survival effect?
Oh, so, you know, we can compute, let's say you have two more samples and you have a gene and it is active in some samples, inactive in others, so you can apply your favorite method, Cox regression, Kaplan-Meier, whatever, to see if there are significant survival differences when the gene is inactivated or activated in a certain population of samples. And you find that when these pairs are lost together, the patient's survival is better. What does that further tell you? That yes, we can. They are indeed synthetic lethal, because whenever if they are truly synthetic lethal and they are lost together, the tumor is more vulnerable, gets killed more, and the patient's survival as a result is better. So that's where our initial observations that set us out in 2013, dinosaurs were roaming the earth, right? To start studying this topic. We were very excited. We published our first algorithm to do that in Cell in 2014. And since then, we actually have been working very hard for 5 years to actually make it work in patients. And I'll show you what it means to make it work. Okay? Questions up until now? Cool. So where were we? Okay, we can hopefully... Okay, so I wanted to skip a few slides, but this one is important actually. So suppose I identify these synthetic lethal network, the true synthetic lethal pairs, okay? This is a sketch of the network, okay? So now I have a gene and this is its synthetic lethal partners, and I happen to have a drug that targets this gene and whatnot, okay? Now, if this is true, and if I'm correct, if I identify the correct synthetic lethal pairs, then given this drug, I will be able to predict who will respond or who will not, right? How? Because if I take a biopsy from a patient and these synthetic lethal pairs, predicted ones, are all indeed inactivated, that means they are lost, they are underexpressed, they are mutated, whatever, I will predict that such a patient will what? If the synthetic lethal partners are inactivated in the biopsy from his tumor, and I have a drug that targets the partner, and I see that these guys are inactivate, I will predict that such a patient will what? Will likely respond, right? And if not, I will predict that you will not respond. So I get a useful, tractable, important way of testing and validating my predictions. Can I do patient stratification? Can I tailor in a precise way the treatment of patients based on the whole genome of the tumors? Okay, guys? Cool. So that's the general scheme of things. And now I'll walk you through very quickly, hopefully, because time is short, via the major steps of the algorithm that you can ask me. So our starting point is these two papers published last year in Cell, which took 500 cell lines, let's say cancer cell lines, did CRISPRs or shRNA knockdowns. We have the expression. If you think for a moment, and that's what these authors did, although they are doing only single knockdowns, because they have the transcriptomics, you can infer an initial estimation of the synthetic lethal pairs in cancer. Why? Let's say you are looking at gene A and B. You knock down gene A in 500 cell lines, okay? Go look at these papers. They're quite fascinating. You knock down, if you're interested, of course, you knock down gene A. Now, you divide the 500 cell lines to, let's say, the top 100 cell lines where gene B is highly expressed, and the bottom 100 cell lines where gene B is down expressed, okay? Now, if you see a difference of essentiality, the knockdown of gene A is much more essential in the cell lines where gene B is downregulated than in the cell lines where gene B is upregulated, you can deduce what? That it's a likely SL candidate, right? That's what these guys did. Okay, so by doing this analysis, they come up, depends on your thresholds, what you choose for hundreds of thousands candidate synthetic lethal pairs. Yes, please. Excuse me? Right, so there are tools to infer the functional effects of mutations. Okay? Yes, there are many tools to do that. They're not great, but usual computational whatever accuracy. But for our case, these tools, mutations are not really relevant because they occur mainly very highly skewed in 300, 400 genes. We are relying on copy number, methylation, and expression because we are interested in inferring what's happening on the whole 20,000 genes. Okay? So we do that, and these guys did that, and they come up with a few 100,000 candidates. But actually, when you take these candidates from the cell lines and you infer these criteria, which I intuitively alluded to, right? The occurrence in cancer samples and the effect of survivals, you actually discover that most of these candidate cell lines that they inferred in these two Cell papers are totally rubbish. They're not relevant at all in patient data. There's no evidence that they work in patient data. So what we did is designed on the last few years a new computational pipeline which has 4 steps, which starts from this initial tool, okay? And applies these intuitive considerations that I explained to mine the data. I'm sorry, I have to skip it in the interest of time, but you can ask me. Anyway, it's published, so you can look at the details and apply this and also phylogenetic considerations and not what? To identify, starting from these pools of hundreds of thousands of candidates, a cancer synthetic lethal network, which we call CSL, a clinically relevant network, which depends on your cutoff. In our case, it's about 2,000 genes, 2,000 interactions, something which is manageable, the interactions which are most supported by the tumor clinical data. Okay, and they are enriched in all that you would expect biologically, and now we go on to test them in experiments and in patients' data. And that's what I want to show, and that's what we published recently. So just to skim you through the results, first of all, we collect another independent set of synthetic lethal pairs that were actually tested in the data for a few major oncogenes in double knockdowns, isogenic cell lines, whatever. And we do quite okay. Especially DAISY is this previous algorithm that we published in Cell in 2014. So, and we beat it terribly. So the lesson is that if you're stupid enough to publish so-so algorithms, you have a chance to improve on them a few years later, right? So whatever, so whatever. This is an independent combination, drug response data, whatever. And this is a dream challenge recently. We actually can predict both combinations, both in cell lines and most importantly in xenograft, in mouse xenograft, way better than these random SLs that were inferred from these cell papers or this our lousy previous DAISY algorithm, so on and so on. But this is a bit more interesting. So this dream challenge was about predicting drug response for single agents and synergy in cell line and xenograft data, okay? And there was a competition and a few algorithms won, and these are the top winning algorithms, okay? And we do as good as the top algorithms, okay, that won this dream challenge. This is for singles. For singles, actually, there's one that beats us. For drug synergy, we are better. But we are at the same level. But the key point is, and I cannot emphasize it enough, that these algorithms were machine learning supervised algorithms that trained on the data. We never see the data, the specific data. We train on 10,000 tumor samples. We don't see any drug response data directly in the algorithm. Then we go to an unseen data. We never look at it and we test it and we obtain results that are on par with supervised, most likely overfitting algorithms. Okay. I'll skip that. We predict drug response in the TCGA, not to all the drugs, but to some of the drugs quite accurately. We predict drug response in PDXs. So I want to tell you, we got data from a dominant large PDX company, which, you know what the xenograft PDX business is? So you take cells from patients' tumors. These companies offer a service. They transplant it in mice. They grow it. They try different drugs. And they are looking for drugs that will work. Okay, so it's a nice idea. It has many issues, though. No time to discuss. And it's a costly process and time-consuming. It takes months and whatever. We just took the patient's data. We didn't know at all the drug response data. We didn't grow any mice. We didn't kill any mice. We didn't invest $1, okay? We just took the patient's data, applied our algorithm, predicted which of the drugs will respond best, and in 70% of the cases we predicted correctly the top drug that was found in the PDX without ever, making any mice miserable. And within days, and not within months, and time is of essence.
The top one out of how many?
About 10 to 20, that's what they screen. Right. And finally, in patients, this is a large collaboration, it's still ongoing, so I can't go into detail, with a leading medical cancer center in the East Coast. And we have indications which we can, because we are looking on the whole genome, we can indeed provide a much higher coverage than you can obtain by looking just on a few 100 cancer driver genes. We can predict combinations with immunotherapy, which is a huge field, but I will, since time is running, these are all our many experimental collaborators, but since time, which were really amazing, and you can look in the paper, there's a lot of validations that I didn't cover, new combinations and stuff like that. If you're interested, you can look in the paper. But in the time that has been left to me, I want to address the second problem. So until now, I described to you a method to try and predict, find patient-specific vulnerabilities and attack them based on looking on the whole genome. But there remains another gnawing problem, and that is resistance emergence. And we want to try and do something about that. So to this end, we are identifying and harnessing another type of genetic interaction, which is called synthetic rescues. But let me first explain how currently people are identifying mediators of resistance to cancer treatments. So what are people doing? Basically, what they're doing in each such study, so for each drug where you want to identify the mediators, for drug Y, in cancer type X, you assemble the patients in a clinical trial. You can see millions of dollars already ringing for each drug in each cancer type. You give the drug, some of the patients will luckily respond, others regrettably won't. And then you do your favorite omics data, differential expression, proteomic arrays, whatever. You identify genes that are differentially upregulated in the resistance and not in the sensitive. And then you get a series of candidates. In the good case, you do cell lines or mouse experiments. You knock them down with the drug, and you manage, if you're lucky, to find a few genes that modulate the resistance. Are you with me, guys? Cool. This is great, but has to be done for each drug in each cancer type. And it's very labor intensive. And we came up with a method that, in principle, if we are correct, by mining the patient's tumor data, we can identify the resistant mediators for all drugs in all cancer types without spending $1, and also for new drug candidates. Okay? So how can we do that? It's based on the notion of synthetic rescue interactions. So if you remember, in synthetic lethal interactions, we knocked down one gene, the cells were happy. We knocked down another gene, the cells were happy. We knocked down both, the cells died. Correct? In synthetic rescue interactions, what happens is you knock down one gene, the cells suffer. Then you knock down another gene and the cells are happy. This is called in the literature, and mainly in the yeast literature, suppressor interactions, and has been mapped in the yeast in a genome-wide manner recently. Or, so we call it down-down rescue. You knock down one gene, it's down, okay? And then you knock down another gene and you rescue. We call it DD or down-down rescues. The other thing is that you knock down one gene, and we are interested mainly in knocking down because that's what drugs do. And the cells upregulate another gene or a series of genes to rescue. Okay? This we call down-up rescue. And using a pipeline like the one I described to you before, briefly described to you before, but switching signs, we can mine the 10,000 tumors and identify these rescue interactions. Why? Because intuitively, if you think about it, if the mutation or the loss or whatever of one gene is rescued by the up or down regulation of another gene, these pairs of events will tend to appear in tumors, what? Much more than expected. by the individual occurrence of these events. And their effects on survival will be to worsen survival much more than expected by the individual effects. And actually, statistically speaking, this task, in a way, I can explain, is actually easier than the task of identifying synthetic lethal interactions. And we do that, and notice we are doing something which is quite surprising and non-intuitive. We are identifying resistance events in 10,000 samples pre-treated. So they haven't seen a drug. And yet we identify resistance events. And why so? Because loss events, mutations, copy number, underregulation, whatever, occurs during the tumor evolution, not only by drugs. And we are seeing a vast array of tumors. And for many of these interactions, it happens that we have a statistically significant signal that is good enough to capture them. So that's why we built a pipeline to do that, and we actually go back to these 4 recent Nature, Science papers, whatever, and we can show that we can identify all the modifiers that they did, spending tens of millions of dollars with 80% accuracy without spending $1. Well, if you don't count how you don't amortize the fellowships of the postdocs or whatever. But that's pocket money, right? Yes, please.
Are these resistant genes that you're talking about developing during the tumor evolution process different or similar to the ones that is actually caused by drug resistance? Is the pattern similar?
So yes, because I will show you that can we predict drug resistance using these modulators. Okay, and if you think about it, it makes a lot of sense because these modulators are a component of the genetic architecture of the cells. They are not invented, during the evolution of the cancer cells or during drug resistance. They are there. That's the cell biology. It's just that the treatment or the evolution of the cancer cells capitalizes on them, allows you to expose and identify them if you are attentive enough.
Same resistance is actually caused by one drug treatment that actually causes resistance?
The resistance is an epiphenomena on the natural mechanisms of cells, of cancer cells, that enables them to overcome subpopulations that you see, to overcome and survive genomic events that are occurring during their natural evolution, which decrease their fitness. Something has to compensate for that in the clones that you see that survived in the tumors. So we do that, and whatever. We can identify mediators. We can identify of mTOR. This is some experimental validation that we did, which I will not go in the interest of time. I want to show you this one. So we actually, again with some collaborators, non-small cell lung carcinoma. So people go into effort, they evolve cell lines that gradually they hit them with a drug, and gradually they evolve cell lines over months, sometimes more, of cell culturing that totally are resistant to the drug. And then you can probe it and study resistance and so on. So we predicted, without looking ever at these cell lines, based on mining the tumor data, resistance mediators, okay, of 18 for a drug called DN, whatever, a certain drug. And in 18 cell lines, we predicted 13 rescuers that are down up. That means if you will hit them with other drugs or shRNAs, you will resensitize the cells back to the drug. And in 70%, so we tested that in 18 cell lines for 13 predicted rescuers, and in like 70% of the cases, we were on the mark. That means indeed hitting the predicted rescuers successfully resensitized the cell lines to the original drug treatment. Then we went and inferred the whole network of resistance mediators to all known cancer drugs. So just to explain to you how it looks, you have a drug, and the drug may have one or more targets. So the drugs are these stars, and the targets are these red whatever proteins, and the greens are the synthetic rescues down up of each of the drugs, so that if you hit them, you can increase the efficacy of the drugs. And we did that. And we looked at all cancer drugs in the TCGA and tested our ability to predict the response to the drugs based on the predicted synthetic rescue profiles for each cancer drug in this drug synthetic rescue network. And we compare it to two recent Cell papers again that identified CFEs. CFEs are cancer functional elements, and they claim that they can use them to predict drug response and stuff like that. It's not at all a competition, okay? We even use supervised methods to try and bootstrap and learn based on these CFEs. In most of the drugs, we predict based on these SR modifiers orders of magnitude better than based on these CFE events. The paper is under review. I hope second review. I hope it will soon be out, so you may be able to see the details. Now to something quite disturbing. Since we can estimate the synthetic rescue profiles of each drug, and we have the transcriptomics and the copy number of each tumor sample, we can go back to the TCGA, and for each drug, we collect all the patients that were treated with the drug. We look at their transcriptomics and expression data pre-treatment, and we ask how many of them, according to our evaluations, this predicted SR, how many of them were already resistant to the drugs when they got it in the first time? Okay, this is not adaptive resistance. This is innate resistance. It was hopeless. And you can see that for many of, we are looking at pre-treatment samples. For many of the drugs that patients were treated with, half of the patients, if we are right, were given these treatments where they were already resistant. Now, on a more positive side, we estimate that for many of these drugs that were given, if we will just hit the prime mediator of resistance that we predict, we will double the survival of the patients. Now, many of these are yet impractical recommendations because we don't have the drugs that hit these predicted modifiers. But it's of interest to know, if we are right, that there is a lot of potential here if we hit the drug and concomitantly hit its main resistance modifier. If we are right, for many of the patients, we can double the survival at least. I can explain how it was done. Now, as you know, I'll skip that. I'll just say that, as you know now, perhaps you know that the hottest thing now in cancer therapy, which is really revolutionary, and if you want to ask me, I can tell you more about it, is cancer immunotherapy. So, of course, it's a lot of interest to find combinations of cancer immunotherapy that work better, and I didn't show you the data. This is work in progress, but it's also of interest to hit mediators of resistance to immunotherapy, to identify them, hit them, and see how well our scheme can identify mediators of resistance to immunotherapy. And it's not trivial because we are working with bulk tumor data, and as you know, immunotherapy involves complicated, intricate interactions between populations of immune cells, and I'm sure we can do much better than what we did here. But the initial results are very encouraging. So this is collaborators at MGH in Harvard, unpublished data, melanoma patients. We have the transcriptomics data after checkpoint inhibitors, both for CTLA-4 and for CD1. And we predicted, based on our analysis, not looking on any treatment data, mediators of resistance to these. And then we looked at the transcriptomics data before and after the treatment, and we can check wherever our predictions are correct or not. So just to show you, so we predicted that certain resistance genes will be down up. That means that if you give the checkpoint inhibitor, the cells must upgrade their level, copy number, transcriptomics, whatever. And you can see that for many of our predictions, indeed, for those that we predicted to be up, their expression post-treatment versus pre-treatment was indeed upregulated. And for those that we predicted to be down, because they are DD rescuers, they are rescuers when they are inactivated, indeed the transcriptomics post versus pre was indeed downregulated. And this, you know, the significance of that is very high. And this is for one inhibitor, this is for another checkpoint inhibitor. And what we found actually, that a lot of our predicted mediators in human patients, fresh data from human patients, actually coincide with a recent Nature in paper who identified them in mice. But we actually identify new candidates in humans which were not found in the mouse data, and now with our collaborators we are testing some of these and so on and so on. So to summarize, since I have to end now, I hope I convinced you that there is, it's worthy to look and try to, you know, analyze patients' data genome-wide and harness this concept of genetic interactions based both SL and SR to try and advance cancer treatment. I want to say that what we are doing is really suboptimal. In many ways, what we are doing could and should be improved, and will be. Okay? And there's a lot of ways which I can explain which our work should be taken forward much more significantly. And if done so, I think it's a quite promising way to try to advance precision-based therapy. And I just want to say that the NCI has now opened this new branch for computational biology, data science, and you're invited to look at it. And we have openings at all levels, both for PIs, staff scientists, postdocs. It's not a frequent event where the NCI opens a new branch. It's in what is called the CCR, the intramural division of the NCI. It has a budget of about $2 billion, 260 PIs, research groups in cancer. It's the largest, quiet whatever. And they are organized in about 35 branches. And we will be the computational arm of the NCI. There's tremendous opportunities for collaborators, world-leading groups in cancer genomics, immunotherapy, Some of the leading cancer drugs have been developed there and are being developed there. So if you're interested, go look at these ads. Feel free to email me, and the rest is history. So thank you very much. Any questions? Yes, please.
I'm wondering how come you have enough power to detect associations, the synthetic leafouts in the TCGA data. So I'm just assuming, let's say, 1 in 100 gene is deleted each tumor. So you have so the probability of 2 genes to be deleted just by chance is 1 in 10,000. If you have just around 10,000 samples, I expect to have just one. Just by chance, you will have one deletion of 4 genes. So how can you identify enrichment or depletion of those genes?
Right. So, you know, sometimes where you make, you have a very rational and nice argument, but if your initial assumption is wrong, and so we identify more events as inactivation, and we use not only copy number loss, but also transcriptomics and methylation data and mutation data. And of course, we carefully control for multiple hypothesis testing on the space of 500 million. And we actually remain with, you know, thousands of candidates which we go on to further test. And the details are really just published, so you're most welcome. And if you have questions, you know, just, yeah. But it is surprising. It is surprising, but as Edmund Hillary said, it's there. Right? It's there. It's striking. How can we not look at this data and use it? Right? We have this now opportunity. And you know, we are really riding on a winning horse. Because it took the NCI 10 years, X hundreds of millions of dollars to sequence 10,000 samples. Now they are sequencing 30,000 as we speak in 3 years. If you're sitting there and not going to apply your skills to analyze this data and take advantage of that, guys, shameful. Yes, please.
I have a question about the resistance. Do you think that you can double life expectancy of a patient? But so do you think it can solve the problem, or it's just going to reduce, like it's going to introduce another resistance you'll have to solve in a similar way, or
Yeah.
will it be kind of an ongoing process?
Ongoing. It's a ferocious enemy. I don't know if you know, guys. You know what's the current criteria of the FDA to approve a new cancer drug? You have to show efficacy. Of course, safety and efficacy. What is the efficacy threshold in terms of survival? Do you know? For a drug, $1 billion, $2 billion, 10 years of development, 4 months. 4 months. In general, there are 2 main developments in cancer treatment in recent years. Yes, one of them is precision oncology. The other has been cancer immunotherapy. And it's extremely interesting. I would say that, regrettably, precision oncology as yet doesn't live to its promises, because resistance emerges, wow. It's an evolving population, and you know, there's a lot of efforts being done in combinations and so on, but it's an uphill battle. Cancer immunotherapy, on the other hand, after 20 years of crazy disappointments, the last 5 years have picked up tremendously. And the people who developed it in these 20 years and were, you know, some of them outcasts, are really heroes, in my eyes. And however, we can help a subpopulation a lot, miraculous levels, but only a subpopulation and only in a subset of cancer types. So we need to do better. And of course, all the cancer research and clinical world is looking into that, right? So for example, now they are running hundreds of clinical trials in the NCI. And NCI is in other places, but the NCI is a major hub for running clinical trials. So for us, it's extremely attractive because they are now running clinical trials on the stuff that I've shown you. This is no nonsense. Remains to see if and how it will succeed. Let's hope. But the NCI is now running literally hundreds of trials with combinations of immune checkpoints, shooting everywhere. And most of them are not rationally designed. Trying. We're desperate. Okay? So yeah, if I believe that what we assess is over-optimistic. I don't believe that. But if indeed some of our predictions can double survival time, that's huge. But I think it's too good to be true. I don't believe myself. This is what the estimations show, but probably the reality will be less so. You have to be very careful. A lot of disappointments. And if you talk to cancer researchers, many years in the field, there's a lot of sense of, I don't know how to say it, right? Carefulness. I had some great idea like a year ago, and I thought it's a great idea. On paper, it looked great. And I went to talk, NCI, I went to talk with the person who is #1 in the world, how to fight resistance via pumps. There are various mechanisms of resistance. It's called MDR pumps, multi-drug resistance. And I had some idea how to attack them, whatever, which I thought was great and whatever. He works on this for 30 years and he's the #1 in the world. I went to him. He said to me, yeah, Eytan, you're a nice guy, and on principle, yeah, I agree with you all that, but it will not work. I'm not interested.
All right, let's thank
Thank you.